We developed a rigorous method for isolating cells from human breast milk to study antibody-dependent cellular phagocytosis, or ADCP, by breast milk phagocytes against HIV targets. This technique facilitates the relatively quick and easy isolation of breast milk cells to evaluate the capacity of leukocyte populations to perform ADCP. Demonstrating the procedure will be Alisa, a technician from my laboratory.
After selecting a relevant target antigen, use a commercial kit to biotinylate the protein of interest according to the manufacturer's protocol, and deposit the biotinylated sample into the upper chamber of a spin column. Add PBS up to 400 microliters and cap the chamber before inserting the column into a collection tube for centrifugation. After discarding the flow-through, add PBS to 400 microliters for a second centrifugation. Discard the flow-through, add PBS to 400 microliters, and measure the protein concentration by a spectrophotometer. To conjugate the biotinylated protein to 1 micrometer microspheres, incubate 6 micrograms of protein with 12 microliters of stock beads and 200 microliters of PBS per plate of conjugated beads at room temperature for 2 hours with gentle vortexing every 20 minutes. At the end of the incubation, pellet the protein conjugated beads by centrifugation and discard the supernatant. After gentle vortexing, resuspend the protein in 12